Thank you all so much. So I'm pinch hitting today for one of my MD PhD students. Unfortunately, he couldn't get out of clinic to be here, so I'm presenting his work on my lab's behalf. So I've kind of grown up in the AARS. My first talk with AARS was actually way back in 2005 as a graduate student myself. And so this is my home organization, and I'm just happy to see all of you here. My lab has been studying isotretinoin for some 20 years, and we know that it's extremely effective for acne, right? But can we identify the next isotretinoin? Can we learn what isotretinoin does and then identify the very next therapeutic that works very similarly? Okay. So here's some background on ISO in case you're not too familiar with it, but it's been used for over 40 years. It's a retinoid. It's also known as 13-cis-retinoic acid in the literature. It was FDA approved in 1982. It's a teratogen. That's the main concern with it, but most patients get dry eyes, itch, sun sensitivity with its use. We know that this works. It is highly effective. Over 70% of patients get remission once they've had one course of isotretinoin. So this is the only drug for acne that actually affects each one of the key 4 factors contributing to acne. And so 40 to 50 years of data has shown us that isotretinoin decreases sebum levels, normalizes follicular hyperkeratinization, affects C. acnes levels on the skin, and also modulates the host inflammatory response. But we still don't fully understand how ISO is working when it's in the skin of these patients. So that's a study that we undertook here. Can we learn from isotretinoin to identify the next drug? And so we do this through drug repurposing, and the underlying theme here is that genes, drugs, and disease states are all connected by virtue of their gene signatures. So can we have an isotretinoin gene signature that then can identify the next drug? Can we do this? I wouldn't be standing here if we didn't do it, right? So I'll show you some of our data. But do we really know about ISO before we get to the drug repurposing? And the answer is no, we still hadn't done enough. So we took a step back, and we recruited 18 severe acne patients for our study. And so this is the way it worked. These 18 patients were just going on isotretinoin as part of their standard of care. We enrolled them in our study. We did sebum levels on their forehead, did their lesion counts, did their IgA counts. And then the trick was that these patients had to undergo a biopsy at baseline, so before drug, and then at one time point during the course of their therapy. So we enrolled 18 patients. We did not biopsy all 18 at all time point because 3 to 4 punch biopsies per patient would be astronomically challenging to enroll. And so each patient kind of had one along the course. And then some patients came back 6 months post-therapy for another biopsy. We took these biopsies and we divided them in half. We did bulk RNA-seq, which I'll share with you here, as well as some immunohistochemistry to understand what's going on in the skin. Okay, so first things first, the clinical measurements in our cohort. We see that the IgA score decreases as cumulative dose of isotretinoin goes up. Similarly, the sebum levels go down with isotretinoin. Fantastic. The drug is working as we predict in this cohort, which means we can move to the next step and actually look at our RNA-seq data. So here are a bunch of volcano plots. There's 4 of them here. The very first one is 1 week. The second one is 8 weeks, then 20 weeks, and then 6 months post-therapy. And this is just a very high-level view of what's going on with the data, but I hope you can appreciate that what's going on at 1 week is very different than what's happening at 8 weeks and 20 weeks. And even 6 months post-therapy, there's still some gene alterations in the skin of these patients. So when we look at all of the data together, we performed an intersection analysis to understand what genes are changing and when. And there's only 16 genes that change across all of the treatment time points. And these are the genes listed here. The top 2 that are upregulated are SELSR2 and Nectin-1. These are cell adhesion proteins. We think that they're being upregulated to help normalize that follicular hyperkeratinization. And then all the ones indicated here in blue are all downregulated across all time points. The majority of these are metabolic genes. They're important in metabolic processes. And many of these are also shown in other isotretinoin studies. So we're seeing the same genes come up over and over again. So just looking at a gene list doesn't really tell us a whole lot, so we went ahead and did a gene set enrichment analysis to understand what pathways are being impacted by isotretinoin over time. And so this is shown here, and you can appreciate that isotretinoin is reliably and sustainably suppressing a lot of these metabolic pathways, and those are indicated up here in the blue highlights. We also see that isotretinoin is suppressing androgen response, as well as suppressing mTORC1 signaling across all the treatment time points. So what do we do with this data now that we kind of have an understanding of what's going on? We went ahead and did connectivity mapping. And so to do this, we used the LINCS database. So for those of you that are not too familiar with this, LINCS is a catalog of cellular responses in response to chemical perturbagens. So basically, it's a bunch of cell lines that are treated with umpteen number of compounds. You get a gene expression signature, and then you can overlay these gene expression signatures from the different drugs with isotretinoin and ask what is similar and what is different. And so to do this analysis, we used our 1-week treatment gene set. We compared them with the LINCS signatures. Those that are going to have a positive score are most similar to isotretinoin. Compounds that come up opposite are opposite isotretinoin. We identified 500 so compounds that are opposite of isotretinoin and 308 which are similar to isotretinoin. This is a huge laundry list of compounds to go through, so we actually did a drug set enrichment, right? Trying to understand which compound classes keep coming up. And so these are the most highly enriched compound classes that are most similar to isotretinoin. The ones highlighted in red here are those. Lo and behold, retinoid receptor agonist pops up. This is really reassuring to us, considering that isotretinoin is a retinoid. But we also see an enrichment in mTOR inhibitors, PI3 kinase inhibitors, and tubulin inhibitors. Those drugs that are most anti-isotretinoin are like the glucocorticoid receptor agonists. This, again, makes sense clinically. So we also performed a network analysis to understand where these compounds kind of fit in the grand scheme of things. And so these are the different enriched classes, the compounds here highlighted in gray. And then these are all the compounds that kind of feed into that. And what is highlighted in the red boxes are the actual targets within in the white boxes are the targets within the cell. And so we honed in here on the mTOR inhibitors, because again, they came up in our gene set enrichment analysis, they came up in our connectivity mapping, and they're really enriched here. So what do we know about mTOR signaling in the skin? So we went back to the half of the punch biopsy that we had saved for histology, and we looked for mTORC1 activity. And we did this by doing immunohistochemistry for phospho-S6, which is a marker of mTORC1 activity. When we look at our patient biopsies, so this is the baseline biopsy at 1 week of the patient, so before they're on isotretinoin. And I hope you can appreciate in the bright yellow that there's a lot of phospho-S6 staining, aka mTORC1 activity in the sebaceous glands of these patients. And then 1 week post isotretinoin, or sorry, 1 week on the drug, that level of phospho-S6 activity decreases. When we go ahead and quantify all of this, you can see that there's a downward trend in mTORC1 activity in these patient biopsies, again, supporting what we're seeing with our computational approaches of mTORC1 signaling being involved as a part of the mechanism of isotretinoin. Okay, so what does this mean? Topical mTORC1 inhibitors. Would it be possible to use these as an acne therapeutic? We know that there's already a drug on the market. This is sirolimus. It has been used. It is FDA approved for other indications, aka drug repurposing. It's been used pretty safely, right? It's a good safety profile. There's no serious side effects. If you dig into the literature a little bit, this drug has been evaluated for other skin diseases, EBS, psoriasis, and rosacea, and shows pretty promising results there. And so we will be starting a clinical trial at Penn State here pretty quickly to investigate this particular drug as a potential acne therapeutic. So the take-home points from this is that isotretinoin is our current gold standard for severe acne. It's been on the market for 40 plus years, and it really does work for the treatment of acne. So let's learn from what this drug is doing to identify the next one. All of these omics technologies allow us to do this computational approach to try to understand what else is going on. And then hopefully we can identify safe and effective treatments without that side effect profile of isotretinoin. So MAC is highlighted here. he would do a much better job than me, but he's off in the clinic, so this is his thesis work. We can't do any of this work without our clinical team, my lab team, and our two long-standing clinical collaborators, Diane Thibotot and Andrea Zangline, and it truly does take a village, and AARS has been funding this work for a very long time, so thank you.